Hello, future doctors. Welcome back to GCSE AQA Biology series by Best Medicine. Today we'll be starting with Chapter 4. Chapter 4 includes photosynthesis and respiration. So our topic today is going to be photosynthesis. Photosynthesis is a very high yield topic for AQA GCSE Biology. In this topic, they try to test you occasionally about things that affect the rate of photosynthesis, right? Things that affect the rate of photosynthesis are the kinds of questions that you're going to get in your exams most of the time. One other high yield thing is the equation for photosynthesis. Okay, so let's dive right into it. So photosynthesis, as we already understood from previous studies as well, is a process in which light energy is trapped in the form of glucose and oxygen is released as a waste product. This process is driven by using reactants called carbon dioxide, which enters the plant's leaves through the stomata, and water, which comes through the roots and then the xylem. So let's try to make sense of this. We have water, right? This enters the roots, and from the roots, it's going to go into the xylem, right? And from the xylem, it's going to march all the way upwards to the leaves. And in the leaves, remember what kind of cells we have? In the leaves, we have the top layer of columnar cells called palisade cells, right? Palisade mesophyll. And then, just below the palisade mesophyll, we have another mesophyll that has ear spaces. And that is called the spongy mesophyll, right? So water is going to the palisade mesophyll, the spongy mesophyll, to help in photosynthesis. On top of this, carbon dioxide is also coming in. So carbon dioxide is coming from the stomata, right? Stomata, which is formed by guard cells. Now most of the stomata will be present on the underside of the leaf, and CO2 is going to come in through diffusion into the spongy mesophyll, and more importantly, into the palisade mesophyll. The palisade mesophyll contains the most amount of chloroplasts. So you have this water, right, and carbon dioxide coming in to the palisade mesophyll. Light energy is being used to drive this process, and we're going to store this light energy in the form of a fuel so that we can use that fuel as an energy source later on. Now that fuel that is created is called glucose, right? Glucose. Glucose is a 6-carbon sugar. It's a 6-carbon carbohydrate, and we already learned a lot about glucose in the biological molecules chapter. So glucose formula is C6H12O6. Okay, you need to remember this. Plus oxygen. So that is the entire equation for photosynthesis. Water plus carbon dioxide gives you glucose plus oxygen. Now this is not balanced, so let's try to balance this. Well, we have 6 carbons here, so try to put 6 carbons here. Okay. We have 12 hydrogens here, so maybe put a 6 here to get 12 hydrogens. And now we have 18 oxygens on the left side, so maybe we can put a 6 here to get 18 oxygens on the right side. And this is the balanced chemical equation for photosynthesis. And you need to know this, okay? You need to know this. A question on completing the equation can come in, completing the word equation can come in, the word equation for combined science usually, while the chemical equation for triple science as well as single science. And on top of this, balancing can come in. So I've already showed you how to balance this. 6, right? 6 and 6. And then this is just 1. So photosynthesis is the process by which plants convert CO2 from the air and water from the roots to glucose and oxygen. Now the purpose of photosynthesis is to store light energy into a molecule that can be used as a fuel. And that molecule again is glucose, releasing chemical energy. Light energy is stored in glucose molecules. So because we are storing energy, right? If we're storing energy, we're taking energy from the outside and putting it inside to the reaction, then is that an exothermic reaction or an endothermic reaction? What do you think? Of course, it is an endothermic reaction, right? Endothermic reaction because we're taking in the energy. So photosynthesis is an endothermic reaction. We have Mr. Chemistry Cat here trying to tell us this, because remember, exothermic and endothermic, we see these terms mostly in chemistry, but we're seeing it here in biology. Light is absorbed by a green pigment inside chloroplast called chlorophyll, something we already finished. So chlorophyll is the green pigment that absorbs sunlight, and we already looked at some diseases as well that affect the pigmentation of the plants, of the availability of the chlorophyll. Remember tobacco mosaic virus, right? Rose black spot disease, right? So those were diseases that would cause something called that was essentially cause deep pigmentation. Now, chlorophyll requires magnesium ions to function. Okay, chlorophyll, to make chlorophyll as well, in this structure, in its molecular formula, magnesium is present. So if you don't have magnesium ions, if there's a deficiency of magnesium ions, then you're not going to have what? Chlorophyll, right? And so your leaves are going to be depigmented yellow. And that is called chlorosis. Remember the word? Chlorosis. So the overall balance equation for photosynthesis is 6CO2 plus 6H2O gives you 1 molecule of glucose plus 6 molecules of oxygen. So what are some of the leaf adaptations for photosynthesis? I think we know most of them because we've been studying it for a long time now. But leaf adaptations for photosynthesis. So number one, the leaf shape, of course, right? The leaf is a very wide, usually, right? There are some exceptions in certain plants like pine trees and cacti, but generally leaves have a wide area, right? They're adapted to maximize the surface area for sunlight absorption. Number two, chloroplast distribution. Chloroplast is evenly distributed, especially at the top surface. The palisade cells would have the most chloroplast. Leaf orientation, positioned to optimize exposure to sunlight throughout the day. So leaves have this, it's not like the leaf is hanging vertically down, it faces the sunlight so that most of the sunlight falls on the big face of the leaf. Thinness, it's very thin so that it allows quick diffusion of gases necessary for photosynthesis. And we already discussed this in the gas exchange chapter and the leaf adaptations previously as well. Stomata placement, which is formed by guard cells to balance gas exchange and minimize water loss. Leaf color, because it's got chlorophyll, it's green, so that helps capturing the sunlight. And veins, right? The vasculature. These veins, as you can see here in this leaf, what they're doing is that they're bringing in water through the xylem, and glucose and amino acids, which are manufactured, are allowed to leave through these vasculatures. So veins would contain xylem and phloem tissue. Okay? Now, very, very popular thing that comes for photosynthesis for AQA students is the required practical, which essentially includes a water plant, a pond bead. Okay, you might have seen this if you've ever done past papers. You might have seen this question before, something like this before. But essentially, the purpose of this required practical is to investigate the effect of light intensity on the rate of photosynthesis. Okay, and what we use here is an aquatic organism such as pondweed. Pondweed lives completely in water that helps us study how much oxygen it is producing. How? By trapping the bubbles of oxygen that it produces during photosynthesis. So the required materials for this would be a beaker, a filter funnel, measuring cylinder, pond bead, light source, meter rule, stopwatch. You can also replace this in a different way. You can, instead of using a measuring cylinder, you can use something called a gas syringe. Okay? Occasionally for AQA sciences, measuring the volume of a gas, especially on chemistry and biology, this is a very popular question. Whenever there is a measuring of volume of a gas or anything else, you always want to add the measuring cylinder and gas syringe, okay? Most of the time. So these are very popular options for other questions as well. Freshly cut 10 centimeter piece of pond bead, light source, meter rule, and stopwatch. Okay, so here's the method. You place the pond bead inside the water and place the funnel on top of it. It is completely trapped in water so that you're able to see any bubbles that are being produced. On top of this, you might have a little bit of hydrogen carbonate source here, which is able to source out the CO2 that is required for photosynthesis. Okay, so what's going to happen here is that CO2 is going to go in, right, and water is going to go in to the plant, and then it's going to start producing these bubbles, right, of oxygen on top of producing glucose, which is going to be stored inside the plant. It's producing these bubbles of oxygen. And what we do is we place a measuring cylinder on top of it so that we're able to trap those bubbles. So all those bubbles, all the gas, the oxygen that is produced, it will accumulate inside the measuring cylinder. And we're able to record the volume in a given amount of time. Okay? So what is the independent variable in this particular experiment? The independent variable, remember, is the light source, the light intensity. So we can either change the light source, or we can place it at a given distance and keep on increasing the distance. So for example, we can place it at 5 inches or 5 centimeters, right? And then we can increase it to 10 centimeters, and then we can keep on going and make it 20 centimeters and so on. Changing the light intensity would help us determine how it affects the rate of bubbles being produced, or the volume of gas that is being produced um, in a given amount of time. So, for example, we could have something like make a table here for our recording. So we could have something like a distance of bulb from pond bead. So it could be like something like 0.5 meters, right? 1 meters, 1.5 meters, and 2 meters, right? And then the volume of oxygen or gas collected. Volume of gas that is collected in the measuring cylinder. And then you could have something like, so the closer it is, of course, the greater the light intensity. So you'll have something like maybe 5 cm cube, right? And then this is also maybe very close to 4.5 cm cube. And then this one is probably 4 base, so something like 3 cm cube. Now we get 2 cm cubes of gas. So the further away we go from the pond bead, the lesser the photosynthesis, the lesser the oxygen that is being collected. Okay? Very, very simple. Another way to set up this experiment, and they can, of course, chain this, right, to test you if you're actually able to understand the things and you're not just memorizing things off of the content. They can chain this a little bit, and they can put give you, instead of a filter funnel, they can put it in a test tube, right? And then they can, instead of attaching a measuring cylinder, they can attach a gas syringe. But the experiment is pretty much the same. In this particular example, we also have the hydrogen carbonate solution, which is a source for CO2. This will provide the CO2 here. Water is already there, and it's going to start producing these oxygen. And then oxygen is going to be collected in the syringe. And again, you can put a lamp here and try to change the light intensity by moving the lamp away or closer to the pond bead, right, using a ruler, and then record your data in a table like this. Okay? So again, the method is to record the distance of the bulb and the pond bead to find out the volume of gas being produced in a controlled amount of time. Okay? Maybe in 5 minutes, how much volume of gas is being produced at 1 meters or 2 meters or 3 meters of the bulb being away from the pond bead. Now, what are the controls of this? Well, the controls would be anything that affects the rate of photosynthesis. Okay, anything that would affect the rate of photosynthesis. For example, if you have the pond bead that is a greater mass, or that has a greater size or greater amounts of leaf surface area, then that's going to increase the photosynthesis rate, right? So pond bead size, you need to control it. Maybe use the same pond bead to repeat the experiment. Number 2, temperature. Remember, temperature affects the rate of enzyme function, right? So increasing or decreasing temperature would affect the rate of enzymes that are involved in photosynthesis. And so changing temperature would affect or influence the rate of photosynthesis. So you want to control this. Pond bead size and time, of course, because if you give it more time, then more gas is going to be collected, right? Less time, less gas is going to be collected. What is the independent variable in this? The independent variable in this is the distance from the light source. And dependent variable would be the volume of gas collected by gas syringe or measuring cylinder. Do you remember how to make a graph? The x and y axis graph? Remember, on the x-axis, you always have the independent variable. And the y-axis, you always have the dependent variable. Okay? So I think if I were to develop this experiment, I think the volume of oxygen would be as it will be on the y-axis, and distance from the light source is going to be here. And so the graph would look something like this. The greater the distance, the lesser is the rate of photosynthesis. Now remember, the greener the leaf, the more chlorophyll it has. Well, dark green leaf plants, they grow well in dimly lit areas because darker it is in terms of greenery, it's going to have a greater content of chlorophyll. So it's going to be able to absorb more sunlight per unit volume as compared to light colored leaf plants. So light colored leaf plants, they only grow well in good light conditions. Okay, and we get our first meme. So sunlight exists in plants. Oh, this is some serious gourmet stuff. It's the only thing that they consume all the time anyways. Never get tired of it. If I eat the same food twice, I cannot eat it a third time. How are plants even able to do this over and over and over again for millions of years? Okay, what can affect the rate of photosynthesis? Number one, of course, we've talked about light intensity, right? That was the entire experiment of the pond beet. Changing the light intensity, right, would change the rate of photosynthesis. Higher light intensity would mean there's a higher rate of photosynthesis because light is the driving force, the driving energy to store the energy in glucose molecules. CO2 is a reactant, of course, right? So of course, if you increase reactant concentration, then generally it would lead to higher rates of photosynthesis. What about temperature? Remember, temperature affects the rate of enzyme function, so optimum temperatures would be best. If you increase the temperature too much, then it's going to denature the enzymes. If you decrease the temperature too much, then it's going to decrease the frequency and the rate of collisions and decrease the number of molecules that have the activation energy, essentially decreasing the number of successful collisions and thereby decreasing the rate of photosynthesis. So temperature, you want to have the optimum temperature. You don't want the temperature to be too high. You don't want the temperature to be too low. Water availability. Of course, water, again, is a reactant, right? Adequate water is necessary for photosynthesis. Drought conditions can slow down or halt photosynthesis, okay? Chlorophyll concentration. Of course, chlorophyll, again, is the means by which we're able to absorb light. So if you cannot absorb light, then photosynthesis would be decreased. Nutrient availability, essential nutrients like nitrogen, phosphorus, magnesium especially, right, which helps make chlorophyll, and potassium, which helps drive a lot of the enzyme actions, the chemical reactions are needed for the synthesis of chlorophyll and other vital compounds involved in photosynthesis. Okay, so all of these affect the rate of photosynthesis. Now, there's multiple graphs that you need to be aware of and how the rate of reaction can change by increasing or decreasing some of these components. So we're gonna make some graphs here for light, CO2, temperature under this space. Okay, so the first graph I want to make is in which we're going to change the light intensity. Okay, so here's the rate, and remember how we record the rate? There's multiple ways, but one of the most common ones is to be able to determine how much oxygen is being produced. So maybe we're just counting the amount of oxygen that is being produced per unit time, or in a given amount of time. Whatever it is, here's the rate on the y-axis. On the x-axis, we'll have the independent variable. For example, in this time, we're just going to have light intensity, okay? So we're going to be increasing light intensity. So for example, we will have something like light intensity increase, right? So light intensity is increasing. And then eventually, at some point, further increasing light intensity does not increase the rate, right? So initially, increasing light intensity increases the rate of photosynthesis. But then a point comes at which further increasing, maybe here, does not increase the light intensity anymore. So what's going on there? What's the problem there? Well, in this case, from this point onwards, light is no longer the limiting reactant. Okay, light is no longer the limiting reactant. Let's say we're conducting this this particular experiment at 25 degrees Celsius. Okay, low temperature, room temperature, and we're conducting this experiment at around 0.05 CO2 in the atmosphere, in adequate water. If we increase the light intensity, photosynthesis increases until this point. At this point, we say that the light is no longer limiting reactant, right? Something else is the limiting reactant. Light is no longer limiting. Okay? So maybe we can change one of the other components, like the temperature or the amount of CO2. Let's try to increase the temperature. So maybe we can increase the temperature. So we take this graph and we're going to start increasing the temperature. For the temperature graph, we're going to conduct the same experiment at 35 degrees Celsius. Let's try to do this. So in green here, here's the same experiment at 35 degrees Celsius. Okay? So it reaches a higher maximum rate of photosynthesis at 35 degrees Celsius, while at being at 0.05% CO2. Okay? Now let's try to conduct this experiment at 45 degrees Celsius and see if it changes anything or not, okay? Or 40 degrees Celsius. So here's 40 degrees Celsius, right? It's not changing all that much. So why is it not changing all that much? Maybe temperature is no longer the limiting reactant, right? So maybe this is 45 degrees Celsius, 40 degrees Celsius, sorry. 45 would be, I think, too high. Might denature some enzymes. 40 degrees Celsius, right? And 0.05% CO2. So in this case, I probably assume that temperature is probably not the limiting what's limiting the concentration here, or the rate of photosynthesis here. So it could be CO2, right? Because we haven't changed this. CO2 is still 0.05. So I keep the same conditions, but now what I do is I increase the CO2 concentration. And this time, when I increase carbon dioxide, the rate just goes up quite significantly, right? So right now we're increasing light intensity over time, and we're conducting this at 40 degrees Celsius at 1% CO2. And now the rate is quite high. So basically, the conclusion to this is that if you increase any of these components, then the rate is going to increase. If it does not increase further, then that particular component is not the limiting reactant anymore. So for example, light is no longer limiting for the first experiment that we saw here, okay? The second experiment, we saw temperature is no longer limiting. And if you keep on going with CO2, probably CO2 will no longer be limiting at some point. And we'll have to look at some other thing. Maybe the amount of chlorophyll content, or maybe something else. Okay, so that's very, very simple. Quite understandable graph. If you don't really understand this, maybe you can repeat this video a couple of times, okay? And if you still don't understand this, then just let me know in the WhatsApp chat, and maybe we can book a lesson, okay? And next up, we have greenhouses. Do you guys know what a greenhouse is? You guys have probably seen this in nurseries. It's where they keep plants and grow them and culture them in a very controlled and perfect environment. Now, the purpose of a greenhouse structure is to be able to trap the heat, okay? The greenhouse structure is supposed to keep the heat on the inside of the greenhouse. That's where you're growing the plants, to be able to provide the plants a good temperature, a good internal environment, a controlled internal environment for their perfect growth. So how is a greenhouse able to achieve this? Now, this takes us back to a little bit of a background in physics. Okay, so in physics, what you have learned are three modes of temperature transfer, right? In physics, when you do heat transfer, you learn about something called conduction, okay? And number two, convection, currents, and I expect you guys to know this by now, I guess. And the third is radiation. Radiation, especially basically infrared radiation. So these are the three modes of heat transfer that you learn in physics. If you guys don't know this, and just contact us, maybe we can book a lesson on this and try to help you understand how the three modes of heat transfer work. But today we're going to be focusing on radiation. So if you don't really understand this, maybe you can review a little bit from your book and then try to come back. So remember, radiation, infrared radiation, is a type of wave, electromagnetic wave, right? And electromagnetic waves, right, have this order, right, from a highest wavelength to the lowest wavelength. It becomes very high frequency and low wavelength. So we start with radio, right, and microwave and infrared, and then visible light and ultraviolet and X-rays and gamma rays and so on. So infrared is around here. Now here's the thing. When you have infrared, if you go to the right, the wavelength decreases. Here's the symbol for wavelength. Wavelength decreases and the frequency increases, right? If you go to the left from this point, then the wavelength increases and the frequency decreases. What about energy? The energy, as you go to the right, increase energy. And as you go to the left, there is a decrease of energy. So gamma rays are the highest energetic rays. Radio waves are the least energetic rays. Now the heat radiation, the infrared radiation that comes from the sun to the earth is high energy because it's literally coming from one of the strongest energy sources that is around our earth, right? The sun has high energy infrared radiation that it emits. So if it emits high energy infrared radiation, is it going to have a high wavelength or a low wavelength? Of course, it's going to have a short wavelength. So short wavelength infrared waves come from the sun to the earth. When the earth, just like anything else, becomes warm, the earth tries to emit that radiation to try to lose that heat. And when the earth tries to lose that heat, earth is not a very high energy temperature source, radiation source, so earth is going to produce a long wavelength radiation. Long waves to try to lose that heat. So let's have a look at the greenhouse structure, how that works. So here's a greenhouse, right? And then inside this greenhouse, you have all these plants. You've got all these plants and everything over here, right? And so on. And on top of the greenhouse, you have the sun, which is emitting short or long wavelength? Short wavelength, right? Short wavelength radiation is coming, bringing in the heat to the greenhouse. Now here's the thing. The transparent cover that is present on top of the greenhouse allows short wavelength radiation to go through. So there's a cover here that is made up of a transparent material, and this allows the short wavelength to go through. Okay, so now what's going to happen is that the earth here is going to become warm. And when it becomes warm, then it's going to try to lose that heat by emitting its own infrared radiation. But the radiation that the earth, the ground of the greenhouse, is going to emit is long wavelength, low energy infrared waves. These long wavelength radiations are blocked by the roof and the surroundings of the greenhouse. The greenhouse allows only short waves to come in. Short waves are able to come in, but long waves are reflected back. Well, we wouldn't say reflected back, but we would say not allowed to escape. So they will just exist inside this container, inside this room. and they would not be allowed to leave, increasing the temperature of the greenhouse over time. And later on, when we do the greenhouse effect, which is causing global warming nowadays, it will be very, very similar to what we have learned over here. Okay, the greenhouse effect, which is able to cause global warming, is very, very similar to this. It works on the same principle, except on a global level. Okay, so that's a greenhouse. Now, what are the uses of greenhouse? In your syllabus, there's uses of greenhouse, and you need to know this. So uses of greenhouse, it's not a very high yield topic, right? Photosynthesis is very high yield, but this can show up on the exam, so we need to be aware of this. Uses of greenhouse. Number one, special crop cultivation, right? You're able to grow a variety of crops, including vegetables, fruits, and flowers, providing a controlled environment. So you can extend the growing season. And you can also use this for research and experimentation. Actually, around my university, we have a lot of these being used for research. So scientists use greenhouses for research purposes to study plant growth under different environmental conditions. So they control different environmental conditions in different greenhouses to see plants' reactions to that. Okay? And of course, propagation. If you want to propagate some seeds, seedlings, right, or cuttings, then and you want to give them the perfect sort of a temperature and environment, then propagation is a good use of the greenhouses. Seedling production. Again, raising seedlings by providing them the perfect environment. How do we control the greenhouse? The greenhouse is controlled. The greenhouses are expensive because they need to be controlled, right? Their temperature needs to be controlled. They can keep on becoming warmer, so we want to ventilate them, right? Heat and cool them. And so we have these mechanisms put in. So we do temperature control. We also want to control its humidity by humidifiers and dehumidifiers, okay? Light management by shade cloths and curtains. Watering systems like drip irrigation, etc. Pest and disease management, right? Using insecticides and pesticides. The economics of a greenhouse. Greenhouses are expensive. This is why they're not often used for regular commercial crops. They're just used for special crops that need special attention, or for propagation, or for scientific research. That is because controlling the above mentioned things like temperature, humidity, light, watering system, pest and disease management, it's quite expensive, right? It can use a lot of resources. So therefore, the greenhouses are only used for special plant propagations and cultivations. Okay? For example, for research methods or growing very delicate plant crop species. When you gotta grab the plant from the very back of the shelf in a greenhouse. Hilarious. And this is a meme on global warming. So when the greenhouse effect works a little too good, we're all fried. I was talking about global warming. Okay, and that's it for this video. So hopefully I'll see you guys in the next one, in which we do aerobic respiration as well as anaerobic respiration. So I hope you guys can review these videos a couple of times and try to do as many questions as you can. And yeah, thanks so much for watching. Like and subscribe. Please share this with all of your friends and classmates, because it's free, right? And if you need to book any lessons, just give us a text on the WhatsApp. Adios.